A leaf, half lit by forest sun. An ant, frozen mid-step. It opens its jaws, grips a vein, and never lets go. Hours later, a stalk breaks the silence. It pushes from the ant's body like a periscope of intent. Spores will ride the wind. The choreography has been precise. This is Ophiocordyceps, a fungus that reads the forest as if it were a clock. It senses light and humidity. It feels heat and height. It drives the ant to a perfect underside of a leaf at a precise distance from the ground, where temperature and airflow promise the best chance for bloom. The ant becomes a vehicle, an instrument tuned to a single goal. Scientists call it a death grip, but for the fungus, it's a birth. Inside, fungal threads weave through muscle, sparing the brain just long enough to use it. They release chemicals that alter movement. They take over the jaw muscles, hardening them into a clamp. At the end, the fungus consumes what remains. This is not horror for horror's sake. It's survival written in another language. Parasites manipulate behavior across the tree of life. Plants, fungi, worms, insects, microbes, each has found ways to pull invisible strings. Why do they do it? Because a body is a bridge. A host moves a parasite to the next stage, the next carrier, the next world. And evolution finds the narrowest path that works. The deeper we look, the stranger the tactics become. Some puppeteers need water. Others need sunlight, or the gut of a bird, or the lungs of a cat. They bend fear, hunger, even sex into tools. We see the ant and ask, how far can control go? Into the nervous system? The immune system? Into the very timing of a life? Pause. One story leads to another. From the leaf's underside, the camera rises through the canopy, over rooftops, toward a quiet alley where a whisker twitches and a shadow moves. The stage changes. The strings stay taut. To meet the next puppeteer, follow the scent of a cat. A mouse nose tests the air. Somewhere ahead, danger. Yet it steps forward. Toxoplasma Gondii, a protozoan with a plan, has taken up residence. It cannot complete its life cycle unless it reaches a cat, so it recalibrates fear itself. Infected rodents show altered responses to the odor of cat urine. Some lose aversion. Others display a skewed attraction. Circuits in the brain, tuned by evolution to shout, run, begin to whisper, wait. How? The parasite sits inside neurons and immune cells. It lodges cysts in the brain. It alters signaling, dopamine, inflammation, the chemical traffic of mood and motivation. In rodents, we can measure changed behaviors, map affected regions, and watch an ancient dance between predator and prey rerouted at the synapse. The cat's gut is the final gate. There, the parasite reproduces sexually, releases oocysts to the outside world, and waits for new hosts to stumble past microscopic seeds. Humans are not the target, but we are incidental passengers. We can be infected, often without symptoms. Studies report correlations, subtle shifts in risk-taking, reaction times, certain psychiatric patterns. Correlation isn't control, and the science is careful here. The details are contested, the mechanisms debated. Yet the question persists. How much influence can a microbe have on a mind that tells stories about itself? We search for proof in scent tests, in MRI scans, in the fine print of immune molecules. We find fragments. We argue over cause and effect. Meanwhile, the parasite waits, patient as stone. Pause. In the wild, nothing happens in isolation. When fear softens in a mouse, a cat moves a little easier, and the parasite writes another chapter. But the stage is wider than attics and alleys. It reaches into rivers and fields, into shells that pulse like lures and worms that demand a plunge. Leave dry ground. Follow the water lines. Because some strings end only where the river begins. A cricket stares at its reflection in a moonlit puddle. Something inside whispers, jump. Hairworms, long, thread-like parasites, grow within terrestrial insects, but can only breed in water. When ready, they press a compulsion into being. The host leaps, drowns, and the worm slips free like a ribbon into its true world. In tidal marshes, a small fish shimmers too brightly. Euoplorchus californiensis lines its brain with cysts, not to paralyze, but to embellish. The fish flashes near the surface, jerks, shows off. Birds notice. Predation rates soar. Inside a bird, the parasite finishes its circuit, eggs drifting back to snails and again to fish, an assembly line of appetite. On a woodland floor, a snail's eyes swell into pulsing, candy-colored clubs. Leucochloridium has invaded the eye stalks, turning them into bait that throbs like a worm. Birds strike. Part of the parasite moves on. Part remains, waiting to infect new snails through droppings. A loop stitched with light. Then, in tall grass at dusk, an ant climbs and clamps to a blade where grazing mouths wander. The lancet liver fluke rides into a cow or deer, finds the bile ducts, lays eggs that drift back to the pasture inside droppings that snails eat, and the circle closes. The climb resets each day with the temperature. At dawn, the ant returns to safety, until evening's hue compels it to rise again. There is also the jewel wasp, Ampulex compressa, which navigates a cockroach like a lock. It delivers a sting to the thorax to still the legs, then inserts its stinger into precise brain centers. Venom alters neuromodulators, curbing spontaneous movement while sparing the ability to walk. The wasp leads the roach by an antenna, a leash of silk. An egg is laid. A larva feeds. So many methods. Yet one question. What are the strings made of? We know the players. Ant and fungus. Fish and fluke. Wasp and roach. But the chemistry of control is a darker chamber. Open it, and we step from spectacle into mechanism. Open it, and the story sharpens. Behavior is biology in motion. To reroute it, parasites reach for levers. Some levers are molecular keys. Venoms from parasitoid wasps contain a cocktail, peptides, and amines that reshape motivation rather than muscle tone. They target ganglia that govern grooming, walking, and initiation of movement, dialing drive down without severing control. Some levers are timing. Fungal pathogens that cause summit disease in flies and ants synchronize death with the environment. Their genes pulse with circadian rhythm. They sense light, temperature, and humidity, then trigger a climb, a bite, a pose that spreads spores. At noon, on a stem. Evening, under a leaf. Each species writes its own schedule. In Ophiocordyceps, gene clusters upregulate during manipulation, secreted proteins, enzymes that remodel host tissue, small molecules that likely influence neuromuscular junctions. Muscle fibers degrade in patterns that spare mouth parts and preserve a final bite. The fungus doesn't need to understand an ant. It needs a lever to tilt a jaw. The immune system is another lever. Toxoplasma can change cytokine signals and tweak the balance of neurotransmitters like dopamine. Inflammation becomes instruction. Neural circuits that weigh risk and reward shift their thresholds by a few millivolts, and that can be enough. Elsewhere, parasites alter smell. Malaria parasites change the bouquet of human breath and skin, making infected hosts more attractive to mosquitoes. The vector arrives on cue, ferrying the parasite onward. No mind control here, just an edit to the air. We are learning to read these edits with genomics, metabolomics, neural recordings. We discover small RNAs that flow between host and parasite, exosome messengers carrying orders across species lines. We see microbial communities, symbionts of the parasite itself, shaping the outcomes. Pause. This is the extended phenotype, genes expressing their will beyond the bodies that carry them. Each lever is a testament. Evolution is not a scriptwriter, but it revises relentlessly. Now pull the camera back. What happens when thousands of tiny edits ripple through a marsh, a forest, a reef? What patterns emerge when fear, hunger, and movement shift across entire food webs? Parasites are everywhere life is. They may be the most common lifestyle on Earth. Their reach rivals that of predators in deciding who eats whom and when. In marshes where Euhoplorchus manipulates killifish, infected fish are eaten by birds many times more often than their healthy neighbors. Energy jumps tiers. Nutrients cycle differently. A parasite's choice becomes an ecosystem's rule. Hairworms drag insects to water, exporting terrestrial biomass into streams and ponds. Aquatic scavengers feast. Fish feed on them. The pulse of carbon changes shape. A thread becomes a current. In grazed lands, liver flukes and tapeworms shape herds. Fertility, migration. Snail populations rise or fall under the weight of parasitic traffic. Clouds of parasite eggs drift in river systems after floods, seeding new mosaics of risk. On coral reefs, isopods lodge in fish mouths or gills, altering feeding and movement. In Arctic summers, botflies and warble flies harry caribou to exhaustion, rerouting migrations and reshaping the tundra's grazed patterns. Not all of this is behavioral control, but the boundary between stress and steering is thin. Even vectors obey edited rules. When malaria adjusts our scent, mosquitoes change their flight paths. When a pathogen tweaks a plant's volatile chemicals, aphids and whiteflies arrive on schedule. The field itself begins to whisper. All of this is not villainy. It is biography. Parasites bind food webs with invisible knots, enforcing intimacy across species that would rather keep their distance. They expose hidden dependencies. They force movement through bottlenecks and gates, through cats, birds, water, and wind, paws, and us. Rabies floods mammalian brains with terror and rage, a viral storm that drives biting and spreads itself with brutal efficiency. There is no metaphor there, only a lesson. Change the brain, change the behavior, change the world. We are not exceptions. We are participants, which brings us to the edge of the map. If control is leverage and leverage is chemistry, what protects a mind? What yields first? The neuron? The immune cell? The story a brain tells itself about a choice? Hold those questions. We have one more step. Stand still. Picture a mouse that doesn't run, an ant that climbs, a fish that dances, a cockroach that follows a silk leash into the dark. All of them are themselves, and something else. We like to place monsters outside us. Yet the most unsettling puppeteers are microscopic, natural, relentless. They do not hate or plot. They explore a landscape of brains and bodies, searching for footholds, building bridges. Zombie is our shorthand for a stranger truth. Behavior is malleable. How far can the control go? As far as chemistry can reach. We know some pathways, dopamine tweaks in rodents, circadian hijack in ants, scent edits in malaria. We suspect others, microbial metabolites touching mood, immune instructions shaping decisions. In humans, the grand claims sync or float on data that are still coming. Caution is the work. So is wonder. We have tools now. We can map single neurons lighting like constellations. We can sequence the whispers of genes mid-symphony. We can eavesdrop on immune cells as they argue under a skull. With synthetic biology, we may someday build our own levers to switch circuits with precision, restoring, repairing, or, if we are careless, rewriting. Pause. There is responsibility in knowing that behavior bends. In medicine, we might turn venoms into cures, parasite tricks into therapies that calm seizures or tame inflammation. In ecology, we might protect wetlands by understanding the parasitic tides that already govern them. But mystery remains. Why do some hosts resist manipulation while others fall? How many different molecules can produce the same compulsion? Is there a universal grammar of control, a small set of rules used again and again by fungi, worms, and microbes across the tree of life? In a forest, an ant loosens its grip and falls to the soil, body spent and purpose fulfilled, just as spores search the air for a new beginning. The camera lingers. The leaf trembles, and the question hangs, as old as hunger. When does a choice stop being ours, and how would we know?